Hi everyone, in this video I'll be speaking about the molecular basis of cancer. I will start by talking about epigenetics. An epigenetic is a gene that is reserved, meaning itself doesn't change, but something happens to it. And two changes that could occur to a DNA is DNA methylation and to its associated protein, histone acetylation. Starting with the DNA methylation, it's when we add a methyl group on the DNA using the enzyme DNA methyltransferase. And this methylation to the DNA represses the gene transcription. So when we have cell division or something, we methylate the DNA, so the gene transcription is repressed. On the other hand, in histone acetylation, we're making the chromatin loose, and this by the enzyme histone acetyltransferase, and this promotes gene transcription, which is needed in the body because our body undergoes regeneration and repair, and it needs this proliferation and gene transcription. However, when there is cancer, the cancer hypomethylates the DNA, so it inhibits gene transcription repression to keep the gene transcription ongoing, and it facilitates the histones to continuously promote gene transcription. For example, motility genes in epithelium cells are supposed to stay switched off by methylation. So they're supposed to stay methylated, to stay switched off, to stay in place. When cancer stops the methylation, epithelial cells move around. Now let's talk about gene mutation. Some genes in our body, when they are mutated, they can develop a tumor. We're going to talk mainly about proto-oncogenes and tumor suppressor genes. So normally we have proto-oncogenes in our body, and these are good things that help us in proliferation, regeneration. But if they get mutated, they can turn into an oncogene, which is a bad thing. Because oncogene is an altered gene which accelerates cell growth and cell division. Proto-oncogene could be activated to become an oncogene via various methods, could be genetics, could be mutations, could be promoter addition, and we're going to discuss some of these things in the following minutes. So an example of activating a proto-oncogene to an oncogene by mutation is RAS. RAS is basically a small gene, G protein, which normally is bounded to GDP, and this is its inactive state at rest state. When the cell receives any type of signaling, this RAS binds to GTP, and this activates it. When RAS is bound to GTP, it's activated. It activates a MAP pathway, which is responsible for cell response, whatever cell response, proliferation, differentiation, any cell response. So this is the normal pathway. However, in cancer, mutation happens to RAS, and it's kept in a locked state with GTP. So when it's locked with GTP, it's binded to it, it's always active, we have continuous proliferation. Now we call it an oncogene. Moving to another way of activating a proto-oncogene to an oncogene by promoter insertion, we have an example of a gene MYC. This MYC is a proto-oncogene normally responsible for cell proliferation. In case of cancers, specifically Burkitt's lymphoma, we have a translocation chromosome 8 to chromosome 14. So instead of MYC being on chromosome 8, it's mistakenly in chromosome 14. And by that, it loses its well-controlled promoter and becomes under another promoter which is active. So it becomes an oncogene because it continuously stimulates the cell to proliferate. The next type of gene sets I want to talk about is tumor suppressor genes, and from their name, these inhibit cell growth and cell division. So normally these genes are present to stop the cell division and control the cell division in the body. However, when it's mutated in cancer, we lose control over cell proliferation because the oncogene keeps stimulating it, and there is no tumor suppressor gene to inhibit it. So we're gonna talk about P53 and retinoblastoma protein, and I'm going to mention their mechanisms. Okay, starting with retinoblastoma, think about it as a gatekeeper that closes the gate between G1 and S, so it doesn't allow the cell to move from G1 phase to S phase. So normally it's unphosphorylated and bound to E2F. E2F is a transcription factor, and the transcription factor is important for cell proliferation. It initiates it. So when we need the cell to divide, a mitogen, which could be any transcription factor that leads to proliferation, this mitogen stimulates cyclin D and CDK4 and 6 to join together and form a complex. This complex will go and phosphorylate the retinoblastoma. By phosphorylating the retinoblastoma, it becomes inactivated and E2F is freed. When E2F becomes free, we can have cell proliferation and cell division. Now, when the cell has finished dividing, the retinoblastoma is dephosphorylated again and binds to E2F, so it's stable. In retinoblastoma that happens in the cancer that happens in children, this retinoblastoma protein is lost. So E2F is always free, having no control over the proliferation of the cell. The second tumor suppressor gene that I want to discuss is P53. And you can think about P53 as a guardian, a genome guardian. So in case of DNA damage, for example, UV light, this P53 gets activated. When it gets activated, it stimulates the promoter of another gene, which is P21. When we get more P21, its job is to inhibit the cyclin CDK complex. And cyclin CDK complex is very essential for the cell cycle to be activated and initiated. So now we're inhibiting this complex, we're stopping the proliferation, we're putting the cell cycle into arrest. Why are we doing that? To allow the DNA repair system to do its job, to correct the damaged DNA before we continue proliferation, because we don't want a cell that has damaged DNA and mutated DNA. If the damage is beyond repair, the P53 stimulates the cells or signal the cell to apoptosis. In cancer cells, they lack this guardian, P53, so many mutations in the DNA and many damages occur to it without having this arrest to repair it.